Okay, so let's start. It's 11 o'clock. Please sit down. Please sit down. So it's a great pleasure to introduce today's speaker. And before I formally introduce her, I have one announcement. On 1st of March, that is the first Saturday of the month of March, TIFR will be open for visits for National Science Day. So all are invited to visit TIFR for registration. Please visit our webpage. And now this brings me to formally introduce our today's speaker. Today's speaker is Dr. Kinjal Sangarajka. She has recently completed her degree, defended her thesis, and now she is a Dr. Kinjal.
Thank you.
And she is a local girl. She's a Mumbai girl, and she has been studying, and studying. That's what I gathered since her school days. And so she finished her bachelor's from our Mithibai College.
Yes.
And then went for master's to MS University, Baroda. And then for PhD, she chose to come to TIFR. And there she worked on a very fascinating field in biology, like how exactly the cells communicate and what are the things involved and what do they mean? I mean, do they have any relation to how a person develops or how a mammal develops? Or is there any relation to any disease that person may develop as the time goes, etc., etc.? But more about that, let's hear from her. Kinjal, it's over to you.
Thank you. Screen share. Screen share नहीं हुआ ना?
Go to Zoom. Go to Zoom. Zoom. Start screen share. Please bear with us for the minute. Please. Yeah. So please minimize.
Okay. So, good morning, everyone, and welcome to the talk. I have the mic.
This was screen. This was screen.
I can be loud. So,
Be really loud.
so good morning, everyone. And today's topic is tiny messages and big package, tiny packages and big messages. So, just a brief introduction, how our brain really works. So, if you consider how the brain works, in an entire day, our brain does a lot of work. Okay, from thinking to eating to walking to any thoughts that you have is all processed by your brain. Now, if you consider how many thoughts does a brain run in an entire day, it would be roughly around 70,000 thoughts that a brain just processes. And it's like a supercomputer which is working in your body 24/7 because brain doesn't sleep most of the time. So this is like the fun fact is your brain is like a supercomputer which is working 24/7. Now, this is not just the brain which communicates the entire process because your brain is here at the top of your head. But your functions, daily functions is with your hands, legs, and everything. So you need a mechanism by which your brain transfers the signal to all the body parts, right? So this comes to the nervous system, which is like brain and the brain nerves, which is like a connected system by which brain communicates every signal. Now, if you go into Zoom, like what exactly does play a role in this thing? So there is a cell called neuron, or we call it as a brain cell. So this is how a typical brain cell looks like, which has a cell body, or which is like, it's like a tree. So when you consider a tree on the top has lot of branches. What does that do? That will collect signals from everywhere. And then it has a trunk or axon, and then it has root-like things. So from the root-like things, it then passes on the message to the next neuron. That is how the cells communicate for a brain communication. Now, this is a very much textbook image how a neuron look like. But are we sure does the neuron look like this? So there have been experiments where people try to image these neurons in a microscopes. And this is how these neurons look like. And it is pretty much like the textbook image that it has these branches, it has a big cell body, and it has branches all over. Now, this is how the neurons communicate. Now, any guesses how many neurons are there in our body? Yes, exactly. So our body has roughly 86 million neurons, billion neurons. That's like more stars than the stars present in the Milky Way. So there are a lot of cells inside our existing body. Now, if you want a cell to communicate, how does that exactly work? Now, if you consider electricity passing in a wire, what happens is it just passes directly, right? But what if there is a gap in between? Now, if let's say there is a gap in between the two neuronal cells. Now, if there's a current passing from here to here, this is in the neuronal body. So from here to here, it can pass as an electrical signal. But if there is a gap, it cannot jump from one cell to another. As a result of it, what it has to do is it has to send a package from one cell to other cell. So consider it as a relay race. Like for a relay race, a person is runs from A to B point, but then it has to pass the baton from one person to another. So the neurons pass messages similarly. That from within a cell, it is an electrical signal, but from a one cell to another cell, it passes the package. So for more details, there is a very nice video. Yes.
Can I interrupt you?
Yes, yes, definitely.
I just want to know why cannot be a permanent connection itself? Why do you want the cell one packet to jump to another? I mean, why the system has been designed that way?
So the system has been designed that way because sometimes it's not just one-to-one connection. Sometimes it activates bunch of other cells. So it's like a junction. Sometimes it activates three different pathways together. So what happens is it releases the packages and all the three neighboring cells catch the package in order to enhance the
That's
signal.
right. Now, still catch the packages.
Right.
The package can be transferred from a junction to several points also. But why there is a gap from one neuron to another? That's what I'm trying to understand.
Yes, because if you consider an electrical signal, it will be just like a go or a no go. It's like 1 or 0. You don't have multiple options for it.
Right.
Now, when the packages are released, there are different kinds of packages which are packed inside. So there are like several chemicals which are packed inside. And based on what chemical is packed inside, it will give you another signal. So you have range of 1 to 20, like so what kind of signal you can have. So for example, there are certain hormones like acetylcholine also, which just don't consider the names, but which help you in the fight or fright whenever you get a shock, whenever you are happy. So there are different emotions related to different kind of package which is being released. Because our body is working on a complex network, it needs this complexity within those packages as well. So
That's
that
right. But main point is why do you need to have gap? Still, you can have the different neurons to give different signal. Intensity may be different. That's a different thing. But why there is a need to have jumping system in this neuron system? If you have this.
That's what I'm saying now, because if just an electrical signal is passing, you don't have that complexity, because electrical signals, you can only vary the intensity by which it can pass. But if you have number of different chemicals which passes then the complexity of signal passing is increased. So you need that system is, I think, what I'm trying to say.
Okay, next question.
Okay, so consider this. So zebrafish is one of the model that we study, but consider that the signal is passing through the synapse. Now, what happens over here? This is like an electrical signal passing by. If you go into deep of what things happen like this, so you can have an electrical signal passing from one cell to another, but once it reaches the nerve end, so this is like once a signal is received, an electrical. Okay. Huh. Huh.
And it goes from neuron to neuron.
The signal strength dilutes based on, so there will be number of particles which release from one cell. Now there are like hundreds of particles released from one cell. How many of them are captured by the other cell is also varied. So because it's in a synapse. So synapse is like a free flowing media. So there will be dilution of those particles which are released also, because not all of them are catched by the next cell.
That's a different thing now. So what happens is that there may be a gap between the two cells may be slightly enlarged because of the various reasons. Then also the signal will not be passed.
There will
the intensity will be smaller. So there is a lot of factor which comes in the picture.
Definitely.
Then why do we need to have a gap? If the gap is there, then the question of increase gap or decrease gap also arises.
Mostly in the synapse, the gap is constant. But what happens is there are different cells which act as a quencher. So what they do is they eat up the extra signals also. So I agree, there are mechanisms by which you can modulate the amount of signal which is passed from one cell to another. And I think this is designed in a way because you can very much tightly regulate this system once you have a gap, because you have now a lot of mechanism which particularly focuses on just transferring one signal to another. So we'll go back to this video again. Sorry. No, I just want to play this. Okay. So consider an electrical signal passing through this neuron, and it is going to some X muscle cell. Let's not consider what happens next. Let's just wait on what happens at this one terminal. So what is happening is you will see that at just zoomed out one image, you will see an electrical signal passing. Now, when you go into the details, you have an electrical signal passing through the trunk or the tree branch. You can consider that as an analogy. It reaches the end of the cell. Now, what happens is once it is reached here, since there's a gap, it cannot pass through another system. What happens is there are opening up of channels, which will take all calcium inside the cell. Once there is excess of calcium inside the cells, these tiny packages over here, which is called as vesicles or synaptic vesicles, fuse to the membrane here. As a result, they will release these tiny little chemicals or the neurotransmitters, which are then attached to on the top of the another cell, which has like a receptor. And then it then passes on the signal to the next cell. If you want, I can go through the video again. Yeah. So again, what happens is, this is one neuronal branch, end point of one neuron or brain cell. Now what happens is, these are little branches which conveys it to the next cell. In this case, it's a muscle here. So you will have an electrical signal within the neuronal cell, which passes through and reaches the end of the tip of the cell. Now what happens, there's an excess of charge over here, as a result of which these open up and there will be influx of calcium. And there's calcium goes inside the cell. Once there is extra calcium over here, now the vesicles fuse over here, and then the small packages are released. These are tiny neurotransmitters or chemicals which bind to the receptors on the next cell. And then it activates the signal there, and the signal is now propagated downstream to the next neuronal cell. Yes.
Potassium, it can be anything, magnesium, anything. Why do calcium?
So the electrical signal which passes in one neuron, it is sodium and potassium. So both of them channel is constitutes the electrical signal which is happening inside one neuronal cell. Now what happens when it reaches at the tipmost of the thing, the channels are voltage gated. So it senses the electric current at the gate. So what happens is, once there is an accumulation of charges, lot of potassium and potassium ions accumulate over there. Those channels open up. Now there's influx of calcium. Now calcium, because calcium is there are like lot of calcium stores inside our body also. So it is very much easy for the body to increase or decrease calcium concentrations at a very fast rate, because we need to keep this in mind that this happens at very fast time scales.
Okay, you can answer the question as to why only calcium. Why it chooses only calcium? Why not magnesium? Why not potassium? Why not sodium? There are many radicals.
There are many. That's what I'm saying. There are two reasons.
Nature has selected only the calcium. That's
There are two reasons. One reason is that the signal which is inside the neuron happens with sodium and potassium. So you don't want to confuse what is happening inside the cell and what is happening outside the cell should not be regulated by the single same ion. So sodium and potassium is out of the question. Calcium, because calcium stores, there are a lot of calcium stores within the body. There are like ER, mitochondria and other cell organelles store a lot of calcium. And also there are hundreds of protein which regulate calcium concentrations, either increase that or decrease that. So for enhanced regulation, nature has designed it for that to be a calcium sensor. If you take these channels out and then you do these experiments with magnesium or something which is divalent, similar to calcium, it works. But for our human body or for any body for that purpose, it has been designed for to it be divalent calcium ions. So going into the static image of how these things look like, it's a similar process that inside the neuronal cells, there is there is electrical signal passing by. And these are the small vesicles, which is like a bag filled with several different chemicals or neurotransmitters. Once the signal is received, you have these channels over here, which open up these channels, and you have influx of calcium. That is, calcium is taken up inside the cell. There is a lot of calcium which enters inside the cell. As a result, you have a lot of calcium concentrations here, because of which it then triggers the release. And then you have these different neurotransmitters which are released now. And that is like how the message is passed from one cell to another. But over here, the important time is also that the time at which it releases these packages is very important. Now, when a one signal passes from one to one, yes. When the signal passes from one cell to another, it is traveling literally at 250 miles per hour, which is like very fast signaling. Because consider that your brain has to work at very fast speeds in order to do your daily work. Like living in Mumbai, we also hustle from one place to another really fast, and your brain has to work faster than that for you to achieve your daily. So how does this happen? For this, the system needs to be ready. Whenever you want to pass a signal, the system needs to be ready and in place. As a result, there is something called as docking. Consider this like the ships parked at a dockyard. You have like multiple ships sitting at the dockyard. Whenever you have a signal, you just load and go. So something similar happens, that most of these vesicles or the packages are held very close to the last membrane where it has to fuse. So they have been like sitting there ready for signal to be transferred. As soon as there's a signal received on the top of the cell, there will be transfer of signals, because electrical signal passes very fast inside the cell. It reaches the end of this thing. And since this machinery is already ready, it just releases then and there. And this is also some of the images which is taken from an actual cell, where you can see these tiny little particles held very close at the plasma membrane or the last membrane of the cell. And minute you see the signal, which is now you can see some of them fusing to the plasma membrane, and this happens in milliseconds time scale. Now consider this as a problem. Now you have one package, one synaptic vesicle, and there's another plasma membrane. Both of them have a membrane. Now what is a membrane? Lipid membrane is typically of this conformation, where it has lipid tails or oil-like behavior in the middle, and they have polar heads, which is like water property. Now consider this, that you have a sandwich of oil, water, oil. Water, oil, and water. Now this is one set of membrane. So over here there is water. In the gap over here there is water. On the top of this there is water, and then there's oil. Now you want these two membranes to fuse. So consider that you have two lipid droplets in a sea of water. Now if you want them to fuse, you need to bring them together in order to displace the water in between and then merge together. Because otherwise water in the sea, oil in the sea of water will just displace. It will not fuse because oil generally runs away from water. So now if this is the problem, what you need is you need something to push them together or bring them together. Here comes you need something to bring these two membranes together for order to do this release process. Now this happens with a proteins called as snare proteins. Now these snare proteins are just like in old age days, you had those telephone wires which had just the spirals, long spirals. These proteins individually are those spirals. What happens is, how does this protein work? Is it starts like a zip. It starts interacting from one end to another. What happens is, if consider this like a zip. So you have two proteins at the edge. It starts unwinding, it starts rewinding. And then what happens is, it then brings the two membranes close together. It keeps on zipping, bringing the two membranes close together in order for it to now fuse and release the particles. Now, why at all these proteins are important? Because this entire process happens in just 1 millisecond. So this has to be very fast. And what these proteins were, yes.
Two things. One is we are showing different colors of snare complex. What does it signify?
This. So there are 3 proteins. I will come into details. So snare proteins is like a family of proteins. There are a lot of different proteins, but mainly proteins are of 2 kinds. One is which is present on the vesicle. That is one category. Let's say A category. And there is one which is present on the plasma membrane or the other membrane. So you have 2 membrane components. One part of protein is on one cell, other part of protein is on the other membrane. So these 3 proteins together form this entire bunch of helix because it has to interact. So consider like 2 ribbons, then forming a ponytail sort of a thing, bringing these together. And that is how now the signal is transferred. So that is why the colors are different, because they are from different proteins. So it forms a complex together.
How does that as to where it should be?
So I will come to that. Just the next slide. So the same question, that this is one snare complex. It was discovered back in 1993, for which people got Nobel Prize also, whoever discovered these proteins. And the problem is that these are not just one protein. There are a lot of snare proteins which catalyze this same mixing to membranes at different things inside the cell, outside the cell. So be it anything, your hormonal release, virus fusing to the cell, everything is controlled by these snare-like proteins. Now, how is the specificity reached? Because when you have when the cell undergoes the developmental process, you have very sorting mechanisms. What they do is, generally what happens is, all the proteins are made here in the endoplasmic reticulum, or it is one of the organelle inside a cell. All the proteins are made here, and then you have endoplasmic reticulum, where the proteins are made. Now these proteins have to be packaged and sent to wherever they play a role in. So here, most of these particles are packaged in the vesicles, and then they reach their respective places. That is how the snare complex, which is important to play a release at the synapse, reach there. Again, a question: if you mix 2 non-related snares in a test tube, will they merge together? Yes, it will, because they are structurally very much conserved. They are very much same. If you consider all the snare complexes, they are very much similar. So when you mix 2 of them, 2 separate of them, to some degree, they can mix together. But since there are a lot of regulators and compartmentalism, that one snare only reaches one place, the other snare reaches only the other place. Because of this, there is very much specificity achieved. I hope I've answered your question. Now, going into again the same release, how does this happen? So consider this end of the sermon. There's one package which is now being released. Now, how does the snare proteins look like? So there's one snare on the vesicle, other two proteins on the plasma membrane, and it just brings two together and now it fuses. This is how the proteins work. I can go back to the video. Yes. Yes, I'm just doing that. So consider this cells fusing at the packages fusing. This is one vesicle which has one snare protein, which is like a coil. It mixes with the other coil. It undergoes interactions by which it brings the membrane close together and then it fuses. Because you need a force. It's like a pulling force. Both of them pull the membranes together. So it's a force by which this transfer is happening. Consider that two lipid vesicles, you are purposely colliding them. So you are generating that force because of that unwinding of those, winding of these proteins at the plasma membrane or at the site. You want to see the video again? Okay. Now, this step of two membranes fusing together, it's not just limited to how a message is transferred inside a brain cell. It is important for the virus entry to your host cell or to your body. It is also important in smaller organisms like yeast. It is also important for any transfer of messages either inside the vesicle or outside the vesicle, like releasing hormones, releasing your fighter molecules, the ones which fight the infection. All of these molecules are released inside the bloodstream, and all of these take place because of these vesicle fusion mechanisms. Now, why at all this is important? Now, you must have heard about a lot of diseases like Alzheimer's, Parkinson's, which is a neurodegenerative diseases. What happens, let's say this is Alzheimer's. What happens is it starts with memory loss and then goes to other severe problems where you have to be dependent on other people, and then you have motor impairment, as in your hand movements, your leg movements have stopped working. What happens is, and there's another disease which is Parkinson's disease, where you have tremors or your hands and legs start shaking, you have body posture is gone, and most of them have the neuronal cells being damaged either with age or because there are problems in your entire neuronal circuit or there are problems in your brain cells. What exactly happens is, because of multiple reasons, I'm not saying release is the only problem. There are multiple reasons by which this disease happens. But what happens because of this disease, when your brain cell gets weak, as a result of this, consider you have transfer of signal. Now what happens is, before in a healthy individual, 100 signals are being passed. But when your neuron gets damaged with age or because of other different clinical problems, now 50 signals are being passed or 40 signals are being passed. As a result, the signal which reaches from one step to another keeps on decreasing, because of which now there is memory loss or there is loss of movement. In both these diseases, in this case, it's the acetylcholine, and in this case, it's the dopamine neurons. These are different types of chemicals, but basically it disrupts the reduced neurotransmitters or reduced amount of packages which are now being passed from one cell to another, as a result of which you have weakened signals, and then you have all those major effects that you see in one person also. This can also be interesting to study the release, because now you can target those. So coming back to the same signal where, yes. Quantum physics to cure these diseases. See, quantum physics, I don't know much about quantum physics, but what I can know is all these theoretical studies, you can understand how the thing happens. Correct. You can know the process in much more detail. So what even I am studying right now is just to know how this process is working. So consider these proteins were discovered 30 years back. Till date, we don't know what is the structure. Like if the whole is formed, how does that look? We don't know that. At what time scales does it work? We don't know that. How many of proteins are required to do this function? We don't know that. So what we do with all these different fundamental research problems, be it quantum physics, you use quantum physics, you use chemistry, you use biology, we try to understand this in much more detail. Because of that, once you know every minute detail of this entire process, then you can design medicines which can overcome this, which can target this thing. So it's the same thing that coming back to the same thing. If you know this process, you can develop molecules which block parts of this entire process. So poison also works in a similar way. Poisons block, either it blocks the receptors, or it blocks the way by which calcium enters the body, or it blocks the next neuron where the signals are passed through. And based on these different targets, you can stop the signal or you can enhance the signal. So you can, if you know this process in much more detail, you can now modulate the entire process, either fasten it, slowen it, stop it. You can do multiple things with that. So my entire project was to understand just this one process in much more detail. Now, going into little technical things, I'll be very simple and slow. If you have doubts, you can stop me. How we do it? It's a major question. How we do this entire process? What we do is we make these proteins in the lab. How we can do it? You know the genetic code. So you know that X amount of DNA makes Y protein. What we do is we take that X DNA sequence, add it to a bacterial cells. The bacteria will nicely produce the proteins for us. So we make these snare proteins in the lab. We make artificial packages like this, and we add those proteins to that. So I have two different packages which has the snare proteins that I want to study. Now what I want to do is I want to know at what time scale does these two fuse together. Simple experiment is, what you do is in vesicles like this. So you have two entities with the membrane, and one of the membrane will have fluorescent dyes. So what this experiment is, you have two colored dyes. One is green, one is red. When the red and green dyes come together, because these are fluorescent dyes, so when you excite them or give energy to them at a certain wavelength, it will emit energy at a certain wavelength, or it will give energy at 50, it will release energy at 70. So there's an energy transfer mechanism. What happens is when you have a green and a red dye together, the red dye will absorb all the 70 energy. As a result, you will not see the green color. Consider it that way. Now, when these two molecules are close together, the green transfers the energy to red, and you will only see the red signal. But when these two fuse together, or when there is this membrane fusion process, you will see now the molecules are very much spread, because now it has been fused to a bigger entity. Now it is all mixing together. As a result, the green can no longer transfer its energy to red. So you can see the green color. That is called as spread dequenching. Or you can now see the green color, which is the donor. And then based on that experiments, you can see you can either consider at what time scales this is fusing, because it gives you a proxy of two molecules fusing together or coming together. And if there is a reduction in the signal, you can consider that there are problems in the existing mechanism, because of which now your next process, like your experimental process, is now being altered. But again, this happens at very slow time scales. In our entire body, the signals transfer at milliseconds. Our experimental values only reach till seconds. So we can't consider what is happening inside the cell. For that, there is another experimental setup that has been set up by our lab. So this is called, I mean, I will not go into details of what this is called, but consider it's like there are two chambers. So this is actually how it looks like. It's this tiny. So you can consider there are two chambers. Consider this as for the representation. There are two chambers which has a very small hole over there. The hole is micrometers. Okay, it's 150 micrometers diameter. So it's very tiny. You can barely see it with your eyes. It's just like a needle poke in an entire paper, sort of. It's that tiny hole. There you can paint the lipid molecules and you can have your system. So your one lipid will have your one snare proteins. In this chamber, you add another protein which has your another chamber which has another protein. When these two fuse together, what happens? There will be flow of ions. There will be a connection from one chamber to another chamber. And this is externally connected to a electrical circuit. Now what happens is, once there's a hole or once the fusion of these two membranes happen, there's a hole between the two, because of which it can transfer signal from one to another. And this can be recorded as current. Now, the only benefit of this is because it's electrical signals, you can record it in millisecond timescales. Now, what you can do is you can perturb these proteins. You can introduce some mutations. Now, what are mutations is you alter the existing sequence to something which may cause diseases, may not cause diseases. So there are certain mutations known in the field which are linked to certain diseases. For example, there is a mutation X. I'm not going into details, but there's a mutation X which is linked to schizophrenia patients. So what happens is in the patients, like in either schizophrenia patients or ADHD patients, they have these mutations. But no one knows that what happens because of these mutations. What is it doing actually? What you see in a person is you just see the symptoms, that that person has a memory loss, that person has epilepsy, that person has so and so symptoms. What is happening in the existing molecular mechanism is something that we don't know. If we don't know that, we can't make drugs which target that process. So what we do is we try to mimic the same mutations here and see what happens to this signal transfer mechanism or just the release mechanism. And what we typically see is if we have a signal like this, so it can shift from open to close. Consider that when the hole is open, it is a stable opening. When the hole closes, you will see spikes to the closed state. If this system goes wrong, you can see multiple variations. Either it is just closing, it cannot open. It opens, but as a leaky opening, it doesn't open very strongly. Or it has both, either it remains closed or open. So doing such perturbations, what we try to do in our lab is try to take some of those mutations which are in a disease causing person, try to understand what happens at the release mechanism. And based on that, we can now translate or have some idea of what is going wrong because of which these diseases can occur. I can take questions. I can go back to some of the parts if that is, yes.
Lecture part is over.
It's mostly done, yes.
When you started, you had shown some chips being eaten and then photograph of it. What are the indications there?
No, it is just that your brain does a lot of work.
That's right. Suppose I take a chip, what is really happening that can describe the process?
So when you want to eat, what happens is first your stomach sends signals to your brain that I need food or I need energy. So your signal is being transferred from here to your brain because then your brain receives those signals, processes that, and then sends it to the motor neurons or your hands and legs that you need to do work to get the chips and then eat. So this is like a signal relay which happens from all your sense organs like touch, smell, nose, seeing, sight and everything. So these are all your sensory organs which perceive the entire situation, send signals to your brain processes it and then sends the signal back to your motor neurons, which is mostly in the hands and legs, which does the work and to get whatever you need. So this happens very fast. Even when you're thinking about it, this process is continuously working.
Then you hear the name of Achar.
Hmm.
You just hear the name. You're not done anything.
Yes.
But suddenly your water comes in your mouth.
Yes.
How does that happen?
That is also the same thing. So sometimes your sensory neurons are very sensitive. So what happens is they sense all the information. So if your brain is thinking that I want to eat, immediate.
You just see, you heard the word only. Achar, that's all. You're not done anything.
Huh.
You're not hungry also. So what is the water coming to?
Because even if you don't want to eat, there is always a subconscious mind which is also working, which is like, so some of those actions are not in your hand. Like breathing is not in your control. Your heartbeats are not in your control. So similarly, once you think that achar, or when you hear some words like this, there is automatically salivary glands release saliva in your mind. It's not related that you want to eat or nothing related to that. But there are some things which are like on a feedback mechanism. Like once this thing happens, you need this thing. Similar is the reflex action. Like once you touch something hot, you will immediately move. So there are some actions which are like predefined in a circuit and they are very, very fast in that terms.
So initially the child also knows that something is hot, then only will touch it, then he will experience it.
So,
that gets recorded. But initially when you say that only just hear the name, that means, I mean, what I'm talking is sometimes you program your brain and reprogram your brain. So many doctors are talking about. So the programming which is taking place, that means you are changing the basic design or basic formation of the programming of the brain itself by using the right words.
So what happens is, this is not a new phenomenon. What happens is from the birth of a child, it gathers all the information. Now that person, when the child is small, doesn't know that achar is something that we eat. But with time, he or she may observe around that, okay, achar is food. So there are a lot of behavioral tests in mice also. What they do is they just give, let's say, they give one type of smell and then give food. So mice start associating that once you receive the smell, you will get the food. So similarly, things get programmed in a child also. Like the child initially, of course, doesn't know what achar is, but once that person starts recognizing achar is food, the food process will start once the achar word is heard. It's a similar behavioral test that even in the mice also happens. Like if you give, let's say, one sweet order and related to food, one sound or red light, you show red light and then you prick the mice. Now, once you show the red light, if you train the mice for like 10-20 days, once you show the red light, that mice will start running. Even if you prick that person or not prick that person, it will feel that pain because it has associated red light with the pain. So similarly, you start associating certain words with certain kind of behavior. Yes. So consider this as a closed state. So consider this as an electrical signal. So once the circuit is closed, you don't see any electricity passing or don't see any current passing through the system. Now, once this fusion has happened, now the pore opens up. So now signal is passing or electricity passes from the circuit. So circuit is complete now. So you see flow of ions moving from one chamber to another and then going to the external circuit. Now you see some current flowing through it. That is why it shifts to the open pore. But again, whenever it closes, you again see it to the closed state. Yes, open and close. Huh. Yes, it is not stable. So that's what I'm saying. Na? Ki if consider it ki agar ye system is very much normal. When the system is very much normal, you will want majority of the signal to be released. So it will stay open. So you see it like a stable opening, only one state. But if there is problem with this system, let's say there are mutations, there are some problems because of which there are diseases happening. In this state, you will not see a stable thing. It will keep shifting from one state to another. And as a result, if 100 signals are passing, now only 50 will be passing. And then your signal processing throughout the signal will be. It's a good system, huh? It's a faulty mechanism, you can say that. And you can even consider that this being the most faulty, the second one being the most faulty because it's mostly closed. Like, let's say if 100% signal is passing here, maybe 10% signal is passing here, maybe 50% signal is passing here, and this is somewhere again in the middle. Yes.
Okay, I suggest we'll take one online question.
Yes.
We'll close the online part. Take a tea break. You can again ask the questions to the speakers.
Okay.
So this question is completely from a very different thing, imagined question. Do we know how brain can imagine? How does it actually imagine? Do we have any understanding of that? That's it. I mean, it's completely unrelated to what is present, but I should just ask.
Yes. So, as brain, you consider brain as one organ, but brain has multiple different centers within the brain. So it has multiple different regions which are specialized for some reasons. So for example, there's a hippocampus which is known for memory. So there are different regions inside the brain which are designated for certain regions. And the circuits are imagined there. So imagination is one more center in the brain where it does all those thought-provoking imaginations or deep thoughts which run over there. So it's something that your brain is creating a situation inside your head. It's something like that. So that happens at different centers. It's of course related to signal transfers, but the imagination is also based on your related to somewhat your daily life, and this is something that is not in your control. But I don't.
That's why it is imagined.
That's why it's imagination.
Okay. So I think on this note, we'll thank the speaker, take a break, and then you can again ask the question.
Okay. Thank you.
Thank you. Thank you.
Ask.